This heading, the diversity of life, a phylogenetic survey, it's going to carry us through the next 5 to 6 weeks of the semester. The first few topics in this series of lectures is included on our current unit, but the balance of it will occupy a completely separate unit in the course. But anyway, let's get into this. Look at diversity from a phylogenetic perspective. I want to start off by reviewing the idea of the domains of life. So the domain is a rank in a classification scheme. It is the highest level of classification. Back when I was a student in introductory biology courses, there was no notion of domains. The highest rank was the kingdom. And I completed my intro bio courses at the tail end of a time when there were only 2 kingdoms recognized, plant and animal. So everything was considered to be either in the plant kingdom or the animal kingdom. The kind of big thing on the scene in around 1980 was the recognition of 5 kingdoms. And now we have a rank above the kingdom, and we recognize many more than 5 kingdoms. So it is not that life has changed so much since I was young. It is our perception of the relationships between groups has changed. The whole idea of domains was first proposed by a microbiologist at U of I named Carl Woese. And he proposed this. So Woese proposed the idea of domains out of a need to resolve relationships he discovered between bacteria and a fairly newly discovered kind of prokaryote that was called the archaebacteria. And his solution to what he saw was to propose we need another level above kingdom to account for the patterns of relationship between kingdoms. What Woese first proposed was that the archaea, what was called the archaebacteria, are not a type of bacteria at all. Rather, they are actually more closely related to eukaryotes than they are to bacteria. That archaea and eukarya share a more recent common ancestor than either would with bacteria. So if archaea and eukarya are sister groups, they should share some derived characters, right? That's the most logical assumption. There's a table which is a comparison between the 3 domains of life. So remember, what is really meaningful is shared derived characters, not shared ancestral ones. So for example, if you look at the first characteristic, not having a nuclear envelope, not having a nucleus, that is the ancestral condition. The fact that bacteria and archaea do not have a nucleus is not evidence they're more closely related to each other, because that is an ancestral trait. Some of the important derived characters I want to point out is, for example, that archaea and eukarya have more than one kind of RNA polymerase. That archaea and eukarya both use methionine as the initiator amino acid in translation, in protein synthesis. that introns are present in genes of archaea and eukarya, that histones are associated with DNA in eukaryotes and some archaea. So those are the kinds of things that lead us to the proposal, the view that archaea and eukarya are the sister groups. They are more closely related to each other. They share a more recent common ancestor. So we will take a really brief look at the domain archaea. Some of their characteristics. They are prokaryotic, right? There is no nuclear envelope. The DNA of an archaea is typically prokaryotic, a single circular chromosome mixed right in with the rest of the cell. All unicellular. Archaea are completely one-celled. In fact, all prokaryotes are. Although there are some that are colonial. They form colonies and sometimes very complex communities, but each cell is a separate individual. The cell walls are made out of either carbohydrate or protein. My favorite thing about the archaea, many of them are extremophilic. Philic is to love. They love extreme environments. There are archaea found in conditions that no bacteria or eukaryote could survive. Now, there are some extremophilic bacteria, but there are archaea that are record holders for the kinds of conditions they can survive in. One of the reasons they are more recently discovered is people didn't really think to look for life in some places where archaea can be found. Several years ago, I attended a lecture by Dr. Madigan from SIU Carbondale, who was an expert in archaea. And I jotted these notes down. Some record holders among the archaea. So as far as temperature, there are hyperthermophiles that can withstand temperatures of 113 degrees Celsius. The optimal, just right for them, is boiling water. A psychrophile is adapted for cold environments, so they can withstand temperatures approaching freezing and make a living. Acidophiles, right, that have optimal pHs of less than 1. Alkalophilic prokaryotes are adapted to alkaline environments. Barophiles, pressure, right? Most of these are on the ocean floor, in deep ocean environments. A halophile is adapted to salt environments. There are archaea that live in saturated salt solutions. So it takes some interesting chemistry, some interesting enzyme structure, and modifications to their plasma membranes to survive in these extreme environments. So this is a phylogenetic tree of life. It recognizes the sister group relationship between eukaryotes and the archaea. Each branch here within the archaea would essentially equate to kingdom level diversity. And I want to point a few things out. The relationships between lineages of archaea are poorly understood, right? We have a big unresolved node here, right? Polytomy. They are hard to study. They're hard to rear in the lab because of their extreme environments are adapted to. So we have so much to learn about the archaea, but we know they are a diverse group. I am not going to ask you to learn these names, but rather on the previous slide, I would like you to know what a halophile is, or what is a hyperthermophile. So for now, just know that there are kingdom-level clades and branches on the archaea side of the tree, and some of the more interesting ones are extremophilic. Most of the rest of this lecture, nay, all the rest, is going to be about the domain bacteria. Those are the more familiar prokaryotes to us, and we know a lot more about them. Okay, so the domain bacteria and their characteristics. The first few are common with the archaea, prokaryotic and unicellular. Again, those are ancestral conditions. Almost half the time that life has existed on Earth, everything was prokaryotic and unicellular. One of the unique things about bacteria is the chemistry of their cell wall. It is a combination of carbohydrates and protein that makes up a molecule called peptidoglycan. That is a chemical signature of a bacterium. This is a scanning electron micrograph of bacteria on the head of a pin, at the tip of a pin, right? So, my gosh, they are small. Prokaryotes are typically about 100 times smaller than a eukaryotic cell. Now, hopefully you learned a bit about prokaryotic cell structure back in 101. Let's cover a few of these and some new topics. When it comes to diagnosing bacteria and classifying them, one of the important traits is the shape of the cell. Bacteria can't change shape other than to grow in length. Their cell wall is rigid, and that gives them a very particular options for shape. There are three primary bacterial shapes. We have, from left to right, the spherical shape called coccus, C-O-C-C-U-S. In the middle, bacillus, kind of sausage-shaped. And then spirillum, kind of a wiggly, squiggly bacterial shape. Cocci and bacilli are much more common than spirillum. So it is the rigidity of the cell wall that gives bacteria their specific diagnostic shape. In lab, we're going to learn a staining technique called the Gram stain. And when you stain bacteria with this particular cocktail of chemicals, you will get a reaction where they either turn a dark purple, we call that being gram positive, or a light pink, that is called being gram negative. And that reaction is a function of their cell wall. On the left side of this image, we see a gram-positive species has a very thick peptidoglycan wall, and the chemicals used bind and stain that very dark. On the right, we see a gram-negative species that has a plasma membrane, a thin cell wall, and then another plasma membrane on the outside. That thinner wall and second membrane prevents the dyes from binding as strongly, and it will show up as a light pink. This is a really important diagnostic tool. If you have a sore throat and you get a throat swab, they'll stain it, and if you have gram-positive cocci, you might have streptococcus, right? That's the coccus in Streptococcus is the shape of the cell. So the combination of cell shape and gram reaction can go a long way toward diagnosing what kind of bacteria is involved, if it is an infection, and what kind of antibiotic might be appropriate. This coccus cell also has a very well-developed capsule. The capsule is a layer of carbohydrates, oligosaccharides, short-chain sugars to the outside of the wall, cell wall. This is kind of a cartoony bacterium with an electron micrograph next to it. It's based on a bacterium called Bacillus coagulans. So not only is Bacillus a shape name, but there's a whole genus of bacteria, a big important one called Bacillus. So there we see the layers that might be present from the inside out includes the plasma membrane, a cell wall, and then a capsule, that carbohydrate layer called a capsule. The nucleoid is the DNA, which would be in the form of a single big circular chromosome. Might not look like a circle here, but it's all crumpled up in order to fit on the inside of the cell. Plasmids are really important bacterial structures. So we had our P-Glow lab where we worked with a plasmid. Plasmids are like mini chromosomes, tiny little circles of DNA. This is an E. coli cell where it's been smushed, and all that wiggly squiggly stuff is its chromosome. Then we have some plasmids here, right? Some little wrinkled up circles of DNA. Plasmids often contain genes that render the cell immune to things like antibiotics. Antibiotic resistance is a genetic evolutionary phenomenon, and often the genes that allow a cell to be resistant are found on plasmids. In our P-Glow lab, the ampicillin resistance gene, the beta-lactamase gene, was found on that plasmid. Some bacteria have extensions to the cell. They can have flagellae, one or more. So if a bacterium has flagella, it's mobile or motile, it can swim. There might be short extensions called pili that would allow the bacteria to stick to surfaces, stick to each other, in some cases used for cell-to-cell communication. The bacterial flagellum is convergent to the eukaryotic flagellum, or maybe the other way around. That is to say, the flagellum in a eukaryote is built differently and operates differently from the flagellum of a bacterium, which is rotary. It spins around like a propeller. This is a close-up of the bacterial flagellum. So the proteins are spanning the cell wall, embedded in the plasma membrane. The cylindrical protein turns, twists the flagellum into a rotary action, whereas the eukaryotic flagellum undulates from side to side. So although bacteria lack membranous organelles, they don't have a Golgi apparatus, they don't have smooth ER, some bacteria do have internal membranes, which are infolded serial infolds of the plasma membrane. Here's an aerobic bacterium that has folded membranes full of electron transport chains. There is a photosynthetic bacteria with folded membranes that contain chlorophyll. Essentially, that aerobic bacterium is like a free-living mitochondrion, and the photosynthetic bacterium is like a free living chloroplast, or would be members of the same lineages from which mitochondria and chloroplasts were derived. Endospores are produced by some bacteria. It's sort of a cell in a cell. They'll take their copy of their DNA and package it up inside of a very thick cell wall, so that when that parent cell dies, that highly resistant spore can exist in some cases for hundreds of years, and when conditions are favorable, they can wake up and metabolize and grow and reproduce. All right, next I want you guys to know a bit about bacterial reproduction. It is a lot more complicated than you might have thought. Bacteria and other single-celled organisms can reproduce asexually through binary fission. Make a copy of your DNA, split the cell in half, produce two genetically identical cells. This is what I would refer to as vertical gene transfer. So if a parent cell produces two daughter cells, right? Those daughter cells will grow and copy their DNA and divide into two more daughter cells. Sort of like in genetics when you went from the P generation to the F1 to the F2, genes are being passed down vertically from generation to generation. And in this case, it is an asexual without sex, right? Asexual mode of reproduction. But we're also going to talk about three different modes of horizontal gene transfer, where genes can be transferred between cells that are part of the same generation. One of these techniques is called conjugation. So we see conjugation in this figure. One of the cells is capable of initiating conjugation. It will send out an extension called a sex pilus. And when it contacts the cell wall of another, they will fuse, and that will form a bridge between the cells, and a copy of DNA is transferred from the donor to the recipient. Conjugation is not reciprocal. One cell is acting as the donor, the other as the recipient. This is a very simplified cartoony view where the molecule that's being transferred is a plasmid. So the donor cell reaches out with a pilus, contacts another cell, a bridge is formed. It will take a copy of its plasmid, send it through that bridge. Now both cells have those genes. So for example, if the donor cell was resistant to an antibiotic because of genes on the plasmid, now the recipient cell is too. Here's a slightly more detailed images of conjugation. In A, it shows us a donor cell which has what's called a F plasmid, F for fertility factor. So that cell is F positive. The other one doesn't have that plasmid, so it's F negative. A conjugation bridge is formed. The donor cell will replicate the plasmid, send a single-stranded copy through the bridge. Even as it's being received by the recipient, it will be replicated back into a double-stranded molecule. When the conjugation bridge breaks, both cells are now F positive because they both have the fertility factor plasmid. In frame B, we have a different genotype. It is called an HFR cell. which stands for high frequency of recombination. In this case, the whole plasmid has been integrated into the primary chromosome of the cell. Notice that blue arrow, that's part of the blue segment where it says F factor. That's the leading edge of the molecule that will be transferred across the bridge to the recipient. So there's a spot there where the phosphate sugar backbone is broken. The single-stranded DNA is transferred over. Now, conjugation doesn't last long enough to send the whole chromosome through. So notice in the third frame of that, the conjugation bridge is broken, but only part of the molecule is transferred. So enough to introduce some new genes, but not enough to give the recipient the whole molecule. So in this case, the recipient has picked up a new gene. It's recombinant, but it is not able to initiate conjugation itself. So that's a difference between the plasmid version and the chromosomal HFR version of conjugation. Second mode of horizontal gene transfer is called transformation, where a cell picks up DNA from the environment. It might be a fragment, it might be a plasmid, but if it successfully picks it up and integrates that into its cell, it will be transformed. It can take on a new phenotype because it has brand spanking new genes. And that's what we were attempting in our P-Glow lab. Instead of a fragment, it was a plasmid that was introduced into the environment. So with transformation, you only need one living cell. Conjugation required 2 cells. Here's our third mode of horizontal gene transfer. It is called transduction. And this is a type of transfer of DNA that is made possible by viruses. Bacteria have viruses that affect them. They're called phage or bacteriophage. So a phage virus can pick up a fragment of DNA from one bacterium and inject that into another. The phage is acting as a vector for the transfer of genes. So here we see in this photo on top an E. coli cell that has a bunch of phage viruses attached to it, inserting their DNA. Check out this scenario on the bottom. On the far left, we have a cell that has been infected by a phage. The phage injects its DNA. The phage DNA has hijacked the cell. The cell is copying the phage DNA. It is making the phage proteins. Its own chromosomes being fragmented. When that cell lyses open, some of the viruses that are released have packaged up bits of bacterial DNA instead of viral DNA. That becomes a transducing phage. So instead of hijacking the next cell it contacts, it just injects a bit of bacterial DNA. Bonus, the recipient cell has picked up a piece of DNA from another cell, and it now has a new genotype. There are, unfortunately, bacteria and eukaryotes that make us sick. There are diseases that are caused by germs, right? The germ theory of disease. One of the individuals that help demonstrate this to the world, that sometimes we are sick because of an infection, was Louis Pasteur, which you might associate with pasteurization. He disproved this notion called spontaneous generation, that life can spring forth from non-living material, and again suggested that some diseases could be caused by living creatures, microbes that get into your body and make you sick. This is a famous painting of Pasteur demonstrating how this flask he's holding up in his right hand is all cloudy because of all the bacteria growing in it. The one in his left hand is clear because it is sterile, it's bacteria free. Notice the long curvy neck on that flask. Both of these flasks were boiled to the point of killing all microbes. The one in his right hand was left open to the environment. The one on the left had that swan neck. So when it cooled, moisture accumulated in the curve, kept air from getting into it, showing that microbes aren't spontaneous springing forth, they are being introduced from the environment. Another person who helped show that some diseases are caused by microbes was Robert Koch, a microbiologist shown on the left side of this figure. And Koch developed some procedures or protocols used to prove that a specific microbe caused a particular disease. We call these Koch's postulates. And there are 5 postulates that must be demonstrated to make this cause and effect link. The postulates are shown on this slide. So first of all, each diseased host, you must be able to isolate the same potential disease-causing microbe from every diseased host. And every individual that this set of problems manifests itself, you got to be able to find the same microbe. Then you have to be able to grow that microbe in a pure culture, which can be difficult. Then you take the cultured microbe and you would experimentally inject that into a healthy host organism like a mouse or a rat. And you should see the disease occur in that previously healthy, experimentally infected host. And then you must be able to isolate from the experimentally infected host the same microbe and compare that back to the original sample. So there we have this in a diagrammatic form. So that procedure, or those conditions that must be demonstrated, that's what we call Koch's postulates. So yeah, it's widely accepted that some diseases are caused by microbes. How exactly do microbes make us sick? Well, there are actually several different ways that this happens. In some cases, the microbe might produce a toxin that would interfere with the host metabolism, such as in food poisoning, right? Food poisoning, there's a botulinum toxin produced that screws with your physiology, makes you sick. In other cases, the microbe might be causing physical damage to cells and tissue. This is what happens in malaria, for example. The cells get into your blood and cause blood cells to lyse open and liver cells to lyse open, and it makes you pretty sick. Often, some of the worst symptoms of infection is the immune response that the host is having to the presence of the microbe, like fever. Fever is an immune response. Fever is good in that when your body temperature is elevated a bit, certain trace minerals are sequestered from your blood, making them unavailable to microbes. Your white blood cells are a little more active, but unfortunately, some microbes cause us to have a severe fever reaction, and that high temperature can actually damage tissue and cause a loss of homeostasis. So that is the case with the COVID-19 virus, for example. It causes your immune system to have a really severe reaction. So some of the people that get really sick are getting sick because of inflammation. So that leads to pneumonia, high fever. So yes, unfortunately, some prokaryotes, some bacteria, some eukaryotes make us sick. They're pests. They're critters that spoil our food and make us ill. E. coli, right? E. coli doesn't invoke happy thoughts for most people. The reality is most strains of E. coli are not disease-causing. They're just another part of the microbial community in our guts. In fact, most species of bacteria are actually very beneficial to people and to the natural world. So I want to cover that a little bit more, explain why most bacteria are actually our prokaryotic friends, and talk about why bacteria are actually very beneficial. By and large, they are our prokaryotic friends. All right, so what have bacteria done for me lately? Well, that answer can be divided into two categories. On the global scale, bacteria and other prokaryotes perform valuable ecological services. They are doing things that greatly benefit all of life on Earth. One of those things is photosynthesis. Photosynthesis is the foundation of most food chains on Earth. It is the process where organisms take carbon dioxide and water and use light energy to convert that into organic food molecules like glucose. So, as we'll argue here in a bit, bacteria actually invented the process, and in aquatic ecosystems, in the oceans, in lakes and rivers, almost all the photosynthesis is happening by organisms other than plants. It is algae, which are eukaryotic, and bacteria. So bacteria are very important as laying down the foundation of food chains in aquatic ecosystems. A second ecological service is the way that some bacteria in the soil and water fix nitrogen. So what does that mean, fix nitrogen? I didn't know nitrogen was broken. So fixing a nutrient means you are converting it from a less useful form into a more biologically useful form. 78% of our atmosphere is nitrogen, but that nitrogen is not in a form plants can use. There's a triple covalent bond between two nitrogen atoms, and that is a tough nut to crack. And nitrogen is vital. It's one of the, on the short list of important elements for life. We need nitrogen to make the amino groups for protein synthesis. We need nitrogen to make the nitrogenous bases of DNA and RNA. So nitrogen has to be fixed. It has to be converted into an ionic form that plants can take up, things like nitrate or nitrite ions, and there are bacteria that make a living doing that. They fix the nitrogen and make it available for plants. Plants can incorporate it into their cells. We can eat the plants. It passes on to us. In fact, some plants actually have symbiotic relationships with soil bacteria, plants in the legume family, and they have these little swellings on the roots. That's what we're seeing in this picture, these root nodules. When you look at it through a microscope, there are bacteria living in those nodules. They fix bacteria, and that's a win-win. So if we could try to appreciate this service that bacteria are doing that really mostly goes unnoticed by most people. So yeah, out there in nature, bacteria are doing things like photosynthesis and nitrogen fixation and other processes like decomposition that are beneficial for all organisms on Earth. There's also a list of things that bacteria do much more directly. In fact, what I'm talking about here is the ways in which we humans have harvested, have employed bacteria to do work for us. One of those is that we have within our intestines bacteria that are performing important metabolic functions and making some vitamins available. And they are symbiotic. So the guest is called the symbiont. We have intestinal symbionts that are absolutely required for the health of your digestive system. If you take strong antibiotics to kill, let's say, a skin infection, you may inadvertently knock out a lot of your intestinal bacteria as well. And that opens up ecological real estate for disease-causing bacteria to invade. So it's very important to keep a healthy intestinal flora, as they say. I had big surgery last year, and I was on strong antibiotics, so my doctor recommended that I take some probiotics to help re-inoculate my intestines with healthy bacteria. So each capsule contains billions of bacteria, 30 billion bacteria in each capsule. That's pretty amazing. So that tells you how small these things are. All right, we rely on bacteria very heavily for biomedical research. E. coli, for example, is an important model research organism. We also employ bacteria in the pharmacological industry to make important drugs, hard to synthesize proteins like insulin for diabetics. What this figure shows us is how we could use a bacterium to make drugs for us. So what we do here is extract a plasmid from a bacteria cell, right? And then isolate the chromosome from a human that contains the gene that is the directions for making insulin, and we combine those here. We create a recombinant plasmid that has the human gene in it, and we give that back to the bacteria. And the bacteria are like, hey, DNA is DNA, or whatever. So as the bacteria grow and divide and multiply, they're copying that recombinant plasmid and passing it on to their offspring, and they're actually making that protein. So you can grow bacteria in big vats and isolate the insulin from it and purify it and use it as a therapy. So there are other examples of important proteins that are drugs that humans need to stay healthy that are synthesized by bacteria. One last example of a human service here is the way we use bacteria to clean up our messes. That's called bioremediation. So the top two images are sewage treatment facilities. And the bacteria there are breaking down the waste. So they're often aerated to mix in oxygen to make that efficient. The bottom figure is after an oil spill. All this oil washed up onto the rocks on the beach, And these guys are spraying a solution of bacteria that break down oil as a way of cleaning up that mess. So thank you, bacteria, for your role in bioremediation. All right, now, next I want to talk a little bit about metabolic modes in prokaryotes, prokaryotic metabolism. Bacteria are sometimes referred to as being primitive life forms, and I don't know, that's an overly simplistic view. Yes, they have retained a more sort of ancestral cell anatomy, but from a metabolic perspective, bacteria are quite sophisticated. It's just that most species have a really specific niche, and they perform particular metabolic reactions and make a living doing that. And because they're so small, it doesn't give them the sort of luxury of having the kinds of fancy organelles that we have. But again, they can do things metabolically that we could never do. So kind of think of them as structural minimalists, but metabolic specialists. So here's a nice table introducing some terms relating to nutrition. So we can broadly classify nutrition as being autotrophic or heterotrophic. And autotrophic literally means self-feeding. And what we think of right away in autotrophy is photoautotrophy, using photosynthesis. Taking CO2, or if you're in the water, carbonate ions, as a source of carbon, using light energy to build organic food molecules. That's the kind of metabolism you learned about in Biology 101 in the photosynthesis unit. Bacteria invented it. Algae and plants have borrowed it. So, photoautotrophy. Other autotrophs, though, are chemoautotrophs. So they do not use light as a source of energy. They use inorganic chemicals like iron and sulfur containing compounds. By stripping electrons from them, they can reduce CO2 into an organic molecule. So that's kind of cool. That allows them to be producers and autotrophs in environments where there's no light. In heterotrophy, the most common heterotrophic mode is chemoheterotrophy. That's what we are. That's what E. coli is. Their energy source is organic compounds, glucose and proteins and carbohydrates, right? So they have to get their carbon and their energy from organic compounds. There are, however, photoheterotrophic bacteria that need organic compounds for carbon, but they are capable of capturing light and generating ATP through light. So that's kind of almost a hybrid mode that we call photoheterotrophy. Okay, the final bit of this lecture is going to be a brief look at bacterial diversity. And here again is this tree that shows the 3 domains. This time we're looking at the bacteria domain. And I want to highlight a couple of the really big ecologically and from a human perspective important bacterial lineages. So we're just blowing that up a little bit more. I want to focus on the proteobacteria, the cyanobacteria, and the gram-positive bacteria. So first of all, the gram-positive bacteria would be all those cells that turn dark purple in response to the gram stain, right? That is a lineage. In the lab, we're going to look at the genus Bacillus. That would be a gram-positive organism, part of that gram-positive branch. The proteobacteria. Oh man, that's a really big important one. That would include E. coli, right? So that's an important reason to know that. A lot of the nitrogen-fixing bacteria are in the proteobacteria group. So E. coli, nitrogen-fixing bacteria. This is a proteobacterium. That's important to know about. Rickettsia prowazekii. What is distinct about this is a couple things. First of all, it's a pathogen. It's a disease-causing bacterium. It causes typhus. It has one of the smallest genomes known in a living organism. It only has 834 protein coding genes. It is an obligate intracellular parasite. That middle picture is a eukaryotic cell. You can see the nucleus right here. I'll trace the nuclear boundary. And all this stuff is Rickettsia bacteria living inside that infected cell. This is the closest living relative to mitochondria, which I think is pretty cool. So it's kind of the sister living relative to mitochondria. So presumably the ancestral aerobic bacterium that established a symbiotic relationship that led to the mitochondria came from the same proteobacteria line that Rickettsia is found in today. All right, another lineage that's pretty important is the cyanobacteria. The cyanobacteria we'll look at in lab, a couple of examples. These are, again, photoautotropic bacteria. This is the branch of bacteria from which chloroplasts are derived. They have the same complex oxygenic photosynthesis pathway that chloroplasts do. Chloroplasts were, once upon a time, cyanobacteria. Okay, that's a look at prokaryotes. The next lecture and the last one in this unit will be about viruses, which is covered back in chapter 17 in our book.